I'd like to extend a very warm welcome to our judges. In this video, my team and I will be presenting our project, the Acoustic Wildlife Monitoring of Bats, for the Dell Competition Final Report Submission. To begin with, as we know, bats have often been linked to various viruses that have impacted humans, most recently COVID-19. However, bats are also essential elements of the natural ecosystems that they inhabit, as they are pollinators, and so conservation efforts are also very important. For both these issues, more information is required about bat species and their habitats. The problem arises in the fact that collecting such data manually is complex, as the habitats that these bats live in, caves and rainforests, are very difficult to navigate. Our project aims to solve this information problem. Firstly, we have developed an AI model that can automatically classify 8 bat species based on their audio calls. This AI model is then deployed on the edge in their bat habitats that communicates any detections to our application server, which then presents this information using a user-friendly web portal for our researchers. This can be seen more clearly in this following diagram. Our edge device is connected to a microphone that performs the automatic bat species classification in the habitats and communicates any detections over the LoRaWAN network protocol to our central LoRaWAN gateway. Our LoRaWAN gateway then forwards any messages to our application server through the Things Network MQTT broker. This uses the MQTT network protocol to communicate our detections, as it is event-driven. These detections are then stored in a database and presented to our researchers using the user-friendly web portal. In terms of the innovative aspects of our project, our bat classifier is unique in the fact that it uses audio-based deep learning, Middle Eastern data, and that our performance has exceeded the literature at 97.51% accuracy. Furthermore, the web portal that we are developing has not been done before, and the hardware benchmarking that we have done will help this project be easily extendable. We will now continue with the various aspects of our project.
The edge device, in this case the Raspberry Pi, has two very important devices. Firstly, the Patterson ultrasound microphone, which can detect frequencies of up to 384 kHz, suitable for detecting high-frequency bat signals. It also has an ESP32 LoRaWAN device. The LoRaWAN edge device here is connected to the Raspberry Pi via the serial. Upon the detection of any bat species by the neural network model, it will send a signal to the ESP32 LoRaWAN device, which will then relay this information to the LoRaWAN gateway. We will play a sample sound to see how this data flow happens. Upon the detection of the species, it will send this information to the LoRaWAN gateway. The LoRaWAN gateway can receive information from hundreds of edge devices simultaneously. It has an active internet connection and is also connected to the Things Network's MQTT broker. It is also received by the application server, which is a client to the MQTT broker. If we refresh the page and see the table view, we can see a lot of new detections have been just received from the edge node. These detections also show up in the map view and can be clicked for more information about the detections.
This is the web portal that will be used by researchers for viewing bat detections. On the home page, the user can view a summary of all bat detections on the map. Over here, each bat detection is represented by a color-coded marker based on the bat species shown in the legend. Clicking on a particular marker will display more details of that specific detection in the form of a pop-up. The user can also aggregate this information in the form of a heat map to see the population density of the bat calls. Furthermore, the user can view the same information in a tabular format based on their preference by clicking on this button. The user can also highlight various cities to get an idea of the general location of the detections. This is an optional feature which can completely be removed as well. The user is provided with various filtering capabilities. For example, the user can select one or multiple species that they would like to view the detections for. They can also limit the number of results to be displayed at a given time, or they could use a time range for the detections as well. So if we filter now, we can see the results on the map or in the table, and the header here shows what filters are exactly being used right now. Moving on, we also have a dashboard which provides a graphical summary of the detections in various forms. The pie chart shows the detections for each species as a proportion of all detections. The next graph is a stacked bar chart, which shows the total number of detections per month based on all the years. This will help researchers show seasonal changes over various years. The next graph is a histogram showing the total number of detections over the time scale, and once again it is distributed based on the species. The last graph is a line chart, again showing the total number of detections over time. Here, the user can zoom based on their preference, and furthermore, all graphs have the ability to select or deselect a species to completely add it or remove it from the graph. Lastly, over here, the user can select a species and can view some basic biological information about that particular species. This information also includes an image of the bat and details about the most recent detection for that species. All the information presented in this web portal demo is based on the artificial data used in the MongoDB.
I will now be discussing our progress with our fully supervised AI model. We developed this using Python as well as the Keras and TensorFlow libraries. In the beginning of our code, we initialize our various libraries and load the required data of the various species. We then create our model as seen here. This design was chosen through experimentation and tuning. We then train our model over various epochs, and we then load and evaluate the model. Do note that our method of evaluation is known as k-fold evaluation. This means that for the same model design, we are actually creating 10 instances, training them and evaluating them, so that our final results are averaged, and we know that our good results are not simply random chance, but rather a reliable average over 10 results. Furthermore, we have adopted a method known as smooth, by which we actually expand and balance out any given data by interpolating it and expanding the given dataset. This is frequently used in the case of imbalanced data, which is what we experienced. We then present our results over here, and as we can see, we achieved an accuracy mean of 97.51%. We then generate some other results for presentation purposes, including a confusion matrix by which we can see how often our model predicted a given species and how often that was correct.
Currently, we've managed to deploy the fully supervised CNN model on the Jetson Nano, the Raspberry Pi 3B Plus, and the Google Coral. This required setting up the devices and creating a TensorFlow-compatible environment such as TF-Lite or TensorRT. For the preliminary deployment, the CNN model is made to run for 10 minutes, where the edge device uses the mic to continuously sample the audio and the TF-Lite environment to classify successive 3-second audio segments, and a total of 200 inferences are carried out during this time period. This picture shows the setup environment for the Google Coral. The Coral is connected to the microphone via a USB connection. The serial data connection to the laptop is required to log into the Coral and to run the TF-Lite script for 10 minutes. The power cable is connected to the Coral via a USB Type-C connection. However, the power cable is split in between to create a series connection with a Yocto-amp. Yocto-amp is a device which allows us to accurately measure the current consumed by the Coral or any other edge device over the 10-minute time period.
The results of the hardware benchmarking comparison can be seen over here. We compared the Raspberry Pi 3B Plus, the Jetson Nano, the Raspberry Pi 400, and the Google Coral. Furthermore, for the Jetson Nano, we tried 2 different deep learning frameworks. The metrics used in this comparison were the latency, current, CPU utilization, and the cost of these devices. Firstly, for the latency, we decided that as long as the average latency was less than 3 seconds, which is the window we use when collecting samples, then we consider it to be suitable. Therefore, for this metric, all the devices were considered suitable. Secondly, for the CPU utilization, we decided that as long as there were no choking CPU spikes, which means the CPU utilization reaches 100 and all processes stop, then we decided that this would be a suitable device. As can be seen for this metric, we found that all devices were suitable in terms of having no choking CPU spikes. Therefore, the main comparison we used was the current and the cost of these devices. We found that for both of these metrics, the Raspberry Pi 3B Plus was the most suitable, as it had the lowest current and the lowest cost. Therefore, the conclusion for our hardware benchmarking was that the Raspberry Pi 3B Plus running TF-Lite Deep Learning Framework was a suitable choice for our deployment of our system.
Firstly, the website with a good graphical user interface provides better analytical capability for researchers, and the AI model will make detections more accurate as compared to when done manually. The project can be similarly duplicated for other animal habitats such as birds and insects. Following this, we can also implement a hierarchical deep learning model. First model to find out which type, for example either a bird, insect or a bat, followed by a model which finds out the exact species of the detected type. This draws inspiration from the existing animal kingdom hierarchy. With regards to the social and industrial impact of our project, we firstly aim to increase the information on bat species in a non-invasive manner and also increase public awareness regarding bat population in the region. We also aim to improve ecological conservation efforts and epidemiological studies on bat-related viruses. This will support bat habitat management to reduce risk of viral exposures. We also aim to reduce the bat data collection cost and time to almost zero. Overall, it is an agile setup as it supports remote scientific research where scientists and researchers can view and study detections of bat species in real time on the web application without damage to any natural habitat.